Axicell is the first product, CAR T-cell product, approved for the treatment of diffuse large B-cell lymphoma. I think it was approved in November of 2017. Most of my experience has been with what they now call Kymriah. It started out 5 years ago as CTL019 and then became Tisagenlecleucel, and now they call it Kymriah. And I've only started to use Yescarta outside of clinical research and on a commercial basis, so I have a limited experience. But the efficacy seems to be quite similar to the Kymriah, which I have a large experience with, and that's very satisfying. You know, to think that the CAR T-cell approach is consistently efficacious, regardless of which product you're talking about. Either approach can offer patients that have an unmet need with current therapies a potential therapy that can lead to long-term remissions. And we have long-term follow-up for both Yescarta treated patients and Kymriah treated patients. So I'm very optimistic that both of these products will fill this large space that we have for a new therapy in patients that don't respond to frontline immunochemotherapy for their large B-cell lymphoma. There are differences among the products, however, and you do need to think of them, although they're both T-cell products, they both have receptors for 19, so they attack the same tumor cell target. You have to think of them as different drugs. They have a different co-stimulatory domain on the inside of the receptor. This is a portion that results in intracellular signaling. Yescarta uses a CD28 co-stimulatory domain. Kymriah uses a 4-1BB co-stimulatory domain. And I like to think of those two domains as accelerators that determine the rate of expansion of the T cells, CAR T cells, when they're infused into the patient. The CD28 expands much more rapidly, and the 4-1B is a slower onset of expansion and perhaps persists longer over months as you follow these patients following therapy. So they are different drugs, and there are some differences in toxicities and in how they were developed in terms of production and logistics and lymphodepletion, but prior to administration. But in terms of efficacy, I see them as two drugs which, at about the same rate, between 30 and 40%, are capable of achieving long-term durable responses in patients with relapsed and refractory diffuse large B-cell lymphoma. Although we have two T-cell products which can lead to durable responses, the logistics are different, and these have to be thought of as different drugs. For Yescarta, which is the CD28-driven CAR T-cell, these T-cells that are used to generate the CAR T-cells, the T-cells from the patient, have to be collected by apheresis and sent fresh to the manufacturer for production. So you need to synchronize your collection with the production lab so that they can receive the product and immediately begin processing and producing. In contrast, Kymriah, which is the 4-1BB-driven CAR T-cell, allows us to collect T-cells from a patient or peripheral blood mononuclear cells from a patient and cryopreserve them, and then ship a frozen product to the production facility when it's available to receive it. So I think that offers a little more flexibility, and logistically it's easier because these patients are often unstable. They may be on therapy to stabilize their diseases. There may be a narrow window to collect cells. There may not be a production facility ready to receive them. And if you can collect the cells whenever you can get them and freeze them, and then continue the patient on whatever it is that's keeping them stable, you can then send the frozen product for production. So I think the logistics are easier from the standpoint that we can start out with a cryopreserved product when we use Kymriah. That being said, I suspect that this is historical, and I suspect there will be development of SOPs for cryopreserved cells for being used to generate the Yescarta product, but it's not yet the procedure that's followed.